Thanks for attending the CAPA-sponsored workshop. The title of our workshop is the Optimized Library Generation for Epigenomic Analyses of Ultra-Low Samples. We're going to split this up into three parts. The first part will be a brief introduction to Kappa Biosystems, followed by our guest speakers, both from Emory University. The first will be Benjamin Barwick. He will be discussing his work on DNA methylation, followed by Christopher Scherer, who will discuss his work on ChIP-seq and ATAC-seq. For those of you not familiar with Kappa Biosystems, we are a global life science reagents company, which is focused on evolving enzymes with improved functions for both PCR, qPCR, and next-generation sequencing. Our core competencies are enzyme engineering by the process of directed evolution, the production of high-quality reagents, protocol optimization, application development, and consultative scientific support. As previously mentioned, one of our strengths is high-quality enzyme engineering by the process of directed evolution. Directed evolution is like natural selection in a test tube. Essentially, we use a combination of both random mutation and an artificial selection pressure to evolve enzymes with desired properties. In this example, we start for the wild-type gene encoding for ATAC DNA polymerase. We introduce random mutations by error-prone PCR. The gene library is then cloned into an expression vector and expressed in E. coli. The gene variants are then compartmentalized and expressed in an emulsion. Also in this emulsion, we introduce a selection pressure, where we then screen millions of variants in the presence of this selection pressure. Only improved variants will survive this pressure. We then repeat the process multiple times until we're left with only the best mutants. We then choose the very best to move forward into our product development pipeline. The example shown here is Kappa Cyberfast DNA polymerase. This was evolved to function in the presence of very high concentrations of cybergreen, which is a PCR inhibitor. More recently, we have applied our technology to the creation of next-generation sequencing products. Shown here is our evolution of library preparation, which is expressed in terms of the improvement in library complexity for 1 microgram of high-quality input. When we plot it in this way, the picture starts to look something like this. We see a 10-fold improvement compared to pre-existing library construction workflows with the introduction of our Kappa Library Preparation Kit. This was done by relatively minor adjustments to both reagent formulation and workflow. We saw an additional 4-fold improvement with the Kappa with Bead Library Preparation Kit. This reduces loss during the cleanup steps. And finally, with the introduction of the Kappa HyperPrep Kit, we saw improvements 60 times that of baseline, with specific implications in low-input applications. Our two speakers today have used the Kappa HyperPrep Kit for their low-input epigenomic studies. And with that very brief introduction, I'd like to introduce our two speakers. Our first speaker, Ben, received both his bachelor's and master's at the Georgia Institute of Technology and was formerly trained in bioinformatics. He is currently at the Department of Immunology and Microbiology at Emory University, where he's focused on the role of DNA methylation in development and disease. His current research includes the application of low-input, high-throughput technologies to the understanding of immune response and cell fate. Our second speaker, Christopher Scherer, received both his bachelor's and PhD from Emory University, and he is also in the Department of Microbiology and Immunology at Emory. His previous research focuses on identifying epigenetic mechanisms of T and B cell differentiation, and his current research focuses on improving and adapting sequencing applications for low cell input numbers, including both ATAC-seq, ChIP-seq, Methyl-seq, and RNA-seq. And with that, I'd like to introduce Ben Barwick.
All right, thank you for coming. Kappa provided our travel stipend. So the objectives for today is to basically describe the workflows for low-input DNA methylation analysis, which I'll talk about, then ChIP-seq, and then chromatin accessibility assays that Chris will talk about. So part one is low-input, high-throughput DNA methylation analysis. I'm Ben Barwick. And so why study DNA methylation? DNA methylation is an essential epigenetic modification in mammals and primarily occurs in the context of CpG dinucleotides. It's integral to the regulation of gene expression. It's catalyzed by the de novo methyl transferases, DNMT plus the substrate SAM, or S-adenosine methionine. And what happens is it essentially adds this covalent mark at the 5' position of cytosines. I'm going to represent these methylated cytosines as black lollipops and the unmethylated ones as white lollipops. And so we can use bisulfite sequencing, and we can treat the DNA with bisulfite, and that will deaminate unmethylated cytosines to uracils. And then we can sequence it and determine which cytosines are methylated and which ones are not. And so in my cartoon here, you can see that methylated cytosines at promoters or enhancers are associated with transcriptional repression and silencing, whereas unmethylated promoters and enhancers allow for that gene to be described, and we think of this as a permissive state. And so there are a number of different methods for high-throughput DNA methylation analysis. There are enrichment techniques, and the advantages to these are analyze the whole genome, but the bias and the resolution is poor, so 200 to 500 bases in resolution, and the bias from the antibodies or MBD binding proteins are poor. And the amount of input is typically pretty high. But overall, the cost is reasonable. There are infinium arrays, which are made by Illumina, and these cover less than 2% of the CpG dinucleotides in the genome. Like all bisulfite-based methods, they cannot distinguish between methylated cytosines and further modifications to cytosines such as hydroxymethylation. They're promoter-centric, and they require a lot of DNA as well, typically around 500 nanograms. Perhaps the gold standard in high-throughput analysis is whole genome bisulfite sequencing. This is single base pair resolution, and you can perform this analysis with less than a nanogram of DNA, but the downside is the cost. And so I'm going to talk to you about a modification of whole genome bisulfite sequencing called reduced representation bisulfite sequencing. And the way this works is it will allow us to cover about 15% of the genome at a fraction of the cost, and with small amounts of DNA, as I'll show you. So it takes advantage of various restriction enzymes, and so if we were to look at, like, say, an NEB catalog of various restriction enzymes, you can see that they have a wide range of cut sites throughout the genome. But then when we ask how many CpG dinucleotides are within these or near these cut sites, you can see that 2 of them, TAC1 and MSP1, have a high proportion of CpGs that can be covered. And by digesting the DNA with these enzymes before adaptor ligation, what we're able to do is cover over 4 million CpGs in the human and about 2.5 in the mouse. If you sequence, say, 100 base pairs, which is a little bit longer, the coverage is even improved. And so the workflow is conceptually simple. You start with high molecular weight DNA, and you digest it with MSP1 and TAC1. We split the DNA into 2 components, one about 60% and one about 40%, and we digest them separately. And what this does is it prevents a lot of small fragments from occurring. And so then we mix it back together and clean it up with spry beads. And then at this stage, we spike in a bisulfite conversion control. We use lambda DNA, which is fully unmethylated, so it should all be converted. And we spike it in at a ratio of 1 to 10,000. And so for 10 nanograms of library, that's approximately 1/3 of a picogram. We then end repair and ligate on adapters using the Kappa Biosystems HyperPrep kit. And we synthesize our own adapters from IDT. And I'll explain to you, we use a short adapter strategy, and I'll explain to you why that is through this email that I received from IDT when I first asked for them to synthesize 6 adapters. They sent me a quote, and the quote was $5,000. And so this was cost prohibitive for us. And so we came up with a strategy that allows us to use one short universal adapter. And typically, an adapter is about 60 nucleotides long, and it's fully methylated for bisulfite sequencing so that the bisulfite will not convert the cytosines in the adapter. What we do is we use a short adapter that doesn't have the P5 or P7 sequences and does not have the barcode. We then add the barcode and the P5 and P7 sequences with long primers. And what this allows us to do is essentially use one universal adapter, which is pretty cost effective, around $500 for 200 uses. And then we buy different primers, and we can add the P5 and P7 and the barcodes on. We spend a lot of time making sure that we optimize the adapter ligation part because that's essential for all sequencing applications. And what we found is we titrated in the molar ratio of adapter in non-DNA limiting conditions. So we started with 500 nanograms, and we added 500x, 100x, or 50x molar concentrations of adapter. And in this situation, it didn't really change the library yield. But in DNA limiting conditions, we used 10 nanograms, and when we spike in 500x, 100x, or 50x adapter, you can see this improve the efficiency of conversion. We bisulfite treat using the QIAGEN EPITech kit, and we modified the protocol to have a longer and hotter denaturation step, increasing the efficiency of bisulfite conversion. We then amplify using the kappa uracil-friendly polymerase, and we estimate the number of amplifications by taking a small dilution of the library before amplification, quantitating it with their hyperprep kit. And we typically, depending on the amount of starting material, will amplify between 5 and 15 times. And so we can calculate it beforehand. We know exactly how many times we have to amplify to get a 10 nanomolar library. So what's this data look like? This is an example of a RRBS run. This is looking at the read position and the nucleotide composition. In this case, we only used MSP1. It cuts at CCGG, and the first 3 dinucleotides are CGG. And what you can see, the first one is either cytosine, indicating that it was methylated, or thymine, indicating it was unmethylated. And then the next 2 are GG. This reduces the diversity at the 5' end. This can create a problem with sequencing, and to get around that, we typically pool these with other sequencing libraries such as RNA-seq, ChIP-seq, ATAC-seq. The reproducibility is very good. This is a scatter plot of methylation calls for the same sample that was made with 500 nanograms and 10 nanograms. And you can see that is virtually indistinguishable from the same sample that's analyzed at 500 nanograms and sequenced twice. We pushed the assay even further. We validated this assay using a methyl-sensitive qPCR, and you can see that the data are robust and very validatable. We pushed this assay even further using lambda unmethylated DNA and in vitro methylated 5X DNA. And what we did is we made libraries from 10 nanograms, 1 nanogram, or 0.1 nanograms. And what you can see here, that the correlation is extremely good between 10 nanograms and 1 nanogram, as well as 10 nanograms and 0.1 nanograms. The 5X is fully methylated, that's shown up here in the red dot, and the lambda is almost completely unmethylated, shown down here. And so we have a pretty good assay, and so what can we do with this? Well, as Adriana said, we're interested in B cells and adaptive immunology. B cells are important. They provide antibodies, protect us from pathogens such as influenza. We can model this in the mouse using LPS, which is a bacteria cell wall component that stimulates the B cells. And when we analyze these murine B cells, plasma cells, and plasmablasts, what we could see is that there is an overall decrease in DNA methylation that's shown here on the heat map, with red indicating highly methylated CpGs and blue indicating unmethylated CpGs. And when we quantitate it genome-wide, we can see that there's a progressive decrease in DNA methylation. And we can pick this process apart even further by labeling the B cells with a cell tracker, transferring them into a mouse that doesn't have any B cells and hitting the mouse with LPS, and you can actually pick out those B cells that have not divided at all, shown here. They're stained, they stain brightly for CFSE, which is the cell tracker, and then we can isolate certain divisions. And we did that. We isolated 6 distinct divisions, and we analyzed them by RRBS in biological duplicate, and we also analyzed them for RNA-seq. And by spiking in ERCC synthetic RNAs and using the kappa mRNA stranded kit, what we were able to see is that as those B cells divide many times, you see a global loss in DNA methylation corresponding with a global gain in RNA expression.